Hello students, welcome to EPG Pathshala program. I am Dr. Shoaib Lukman from the Council of Scientific and Industrial Research, Central Institute of Medicinal and Aromatic Plants, Lucknow. Today we are going to talk on the biosynthesis of lipids part 2, which is an inherent part of the paper lipid metabolism. The objectives of today's discussion is to understand the synthesis of branch chain fatty acids. During the synthesis, how do BCKA decarboxylase enzyme acts and what are its implications? Also, in this lecture, we will be discussing about how do fatty acid synthase system works, how it is regulated, and what are its clinical significance. So, the branch chain fatty acids are either typically saturated and are originated in two divergent families. The first family belongs to the iso series of branch chain fatty acids, and the second family belongs to the anti-iso series of the branch chain fatty acids. In this module of the biosynthesis of lipid, we study the synthesis of branch chain fatty acids, how these branch chain fatty acids are synthesized from the acetyl-CoA, and how the branch chain alpha keto acid decarboxylase enzymes work to form the branch chain fatty acid. In addition to this, the fatty acid synthase system operation also be looked into. So continuing with the biosynthesis of lipid, part 2 explain these conundrums. It has been established that actinomycetales include unique branch chain fatty acid synthesis mechanisms together with tuberculostearic acids. It is important to note that these branch chain fatty acid utilizes three important amino acids: valine, leucine, and isoleucine, as a primer for its synthesis. The diagram presented here showed that the valine undergo transamination reaction to form alpha keto isovalerate, which utilizes mannonyl CoA and undergo decarboxylation reaction by branch chain alpha keto acid decarboxylase enzyme to produce 2-methyl propanyl CoA. As a result of decarboxylation, carbon dioxide is released. 2-methyl propanyl CoA elongates in presence of mannonyl CoA fatty acid synthase to form 14-methyl pentadecanoic acid, which is an isobranch chain fatty acid with even number of carbon atoms. Similar to that of valine, leucine also undergo transamination reaction. And forms alpha keto isocaproic acid. This alpha keto isocaproic acid, together with mannonyl CoA, under the catalytic reaction of branch chain alpha keto acid decarboxylase enzyme, forms 3-methyl butyryl CoA. As it is a carboxylation reaction, carbon dioxide is released. This 3-methyl butyryl CoA further elongates to form 13-methyl tetradecanoic acid. This step is catalyzed by mannonyl CoA fatty acid synthetase enzyme system. The final product, 13-methyl tetradecanoic acid, is an isobranch chain fatty acid, but with odd number of carbon atoms. Similar to that of leucine and valine, isoleucine also followed the transamination reaction, resulted into the formation of alpha-keto beta-methyl valeric acid. This alpha-keto beta-methyl valeric acid together with malonyl-CoA forms 2-methyl butyryl-CoA. This reaction was catalyzed by branch chain alpha-keto acid decarboxylase, releasing the carbon dioxide. In the final step, elongation of the 2-methyl butyryl-CoA takes place in presence of enzyme malonyl-CoA fatty acid synthase to yield 12-methyl tetradecanoic acid. It is an anti-iso branch chain fatty acid. So these leucine, isoleucine and valine utilized as a primers for the branch chain fatty acid synthesis of both the iso and the anti-iso series. Similar to that of valine, leucine also undergo transamination reaction. And forms alpha-keto isocaproic acid. This alpha-keto isocaproic acid together with malonyl-CoA under the catalytic reaction of branch chain alpha-keto acid decarboxylase enzyme forms 3-methyl butyryl-CoA. As it is a carboxylation reaction, carbon dioxide is released. This 3-methyl butyryl-CoA further elongates to form 13-methyl tetradecanoic acid. This step is catalyzed by malonyl-CoA fatty acid synthase enzyme system. The final product 13-methyl tetradecanoic acid is an isobranch chain fatty acid, but with odd number of carbon atoms. Similar to that of leucine and valine, isoleucine also followed the transamination reaction resulted into the formation of alpha-keto beta-methyl valeric acid. This alpha-keto beta-methyl valeric acid together with malonyl-CoA forms 2-methyl butyryl-CoA. This reaction was catalyzed by branch chain alpha keto acid decarboxylase, releasing the carbon dioxide. In the final step, elongation of the 2-methyl-butyryl-CoA takes place in presence of enzyme malonyl-CoA fatty acid synthase to yield 12-methyl tetradecanoic acid. It is an anti-iso branch chain fatty acid. So these leucine, isoleucine and valine utilized as a primers for the branch chain fatty acid synthesis of both the iso and the anti-iso series. The branch chain fatty acids synthesizing system make use of alpha keto acids, and these alpha keto acids include leucine, isoleucine and valine, and they use it as a primer or as primers. The branch chain fatty acid synthase exploit the short chain acyl CoA esters as primers. The alpha keto acid primers are derived from the decarboxylation and transamination of leucine, isoleucine and valine to form 3-methylbutyryl-CoA, 2-methylbutyryl-CoA and 2-methylpropenyl-CoA respectively. This 2-methylpropenyl-CoA primers resulting from valine are extended to make even numbered of isoseries of fatty acids like 14-methyl pentadecanoic or the isopalmitic acid. 3-methylbutyryl-CoA primers from leucine is being used to form odd chain number isoseries fatty acids. For example, 13-methyl tetradecanoic acid. Likewise, 2-methylbutyryl-CoA primers from the isoleucine are lengthened to outline the second series of branch chain fatty acid or the anti-iso series, and this series encloses an odd number of carbon atoms such as 12-methyl tetradecanoic acid. The decarboxylation of the primer precursors occurs via the branch chain alpha keto acid decarboxylase enzyme, and these branch chain alpha keto acid decarboxylase enzyme is abbreviately known as BCKA enzyme. In Escherichia coli, which is a gram-negative bacterium, elongation of the fatty acid pursues the identical biosynthetic pathway to produce straight chain fatty acids, where manonyl-CoA is used as a chain extender. The key end products are branch chain fatty acids between 12 to 17 carbon atoms, and their concerto apt to be consistent and trait for several bacterial species. The branch chain alpha keto acid decarboxylase enzyme and the relative activities of alpha keto acid substrates. The enzyme branch chain alpha keto acid decarboxylase is composed of two subunits in a tetrameric structure A2B2, and this enzyme is indispensable for the synthesis of branch chain fatty acids. It is also accountable for the decarboxylation of alpha keto acids produced by the transamination of leucine, isoleucine, and valine, resulting in the formation of primers used for branch chain fatty acid synthesis. The enzyme activity is exceedingly higher with branch chain alpha keto acid substrates as compared to straight chain substrates. In Bacillus species, its specificity is maximum for the isoleucine derived alpha keto beta methyl valeric acid, trailing by alpha keto isocaproate and alpha keto isovalerate. The high affinity of the enzyme branch chain alpha keto acid decarboxylase for the branch chain alpha keto acids assign it to function as the primer donating system for branch chain fatty acid synthase. In this table, we can see the specificity of the branch chain alpha keto acid activity towards the substrate and the product form. As far as for the pyruvate, the activity was 25% with the formation of 4.9 nanomoles of carbon dioxide liberated per minute per milligram of protein, with an Vmax value of 15.2 nanomoles per minute per milligram and a Km of 51.1 micromole, which increases with the substrate ketoisocaproate and ketoisovalerate to 38 and 63%, and then by enhancement in the formation of the product as well as the alpha keto beta methyl valerate is the substrate, which is highly specific, as 100% activity of BCKA was observed with the 19.7 nanomoles of product formed per minute per milligram of protein. There are factors that affects the branch chain fatty acid length as well as the pattern. As we discussed before, the alpha keto acid primers, primarily between 12 to 17 carbons, are utilized to make branch chain fatty acid, and the size tend to be uncertain and uniform among a meticulous bacterial species, which may be changed due to malonyl CoA concentration or heat stable factors or even temperature. So, the malonyl CoA concentration, the stability of heat as well as temperature affects the chain length and the pattern of the branch chain fatty acids. The heat stable factors modify the specificity of branch chain alpha keto acid decarboxylase activity for a specific alpha keto acid substrate, varying the ratio of the branch chain fatty acid produced. It has been observed that an increase in malonyl CoA concentration affect a bigger proportion of carbon 17 fatty acid form till an optimal concentration of malonyl CoA is accomplished. And this optimal concentration is close to the 20 micromolar. In Bacillus species, a decrease in temperature also apt to move the fatty acid distribution vaguely towards 17 carbon fatty acids. The branch chain fatty acid synthase. Bacteria that do not have the aptitude to execute the branch chain fatty acid synthase system via alpha-keto primer. Apply this procedure using short chain carboxylic acids as primers. Distinctive short chain primers consist of isobutyrate, 2-methylbutyrate, and isovalerate. Commonly, the acids required for these primers are taken up from the environment, which is frequently seen in ruminal bacteria. The disparity amid straight chain fatty acid synthase and branch chain fatty acid synthase is substrate specificity of the enzyme converting acyl CoA to acyl ACP. In general, the overall reactions is isobutyryl CoA plus 6 moiety of manonyl CoA plus 12 moiety of NADPH plus 12 proton forms isopalmitic acid plus 6 moiety of carbon dioxide plus 12 moiety of NADP plus 5 molecule of water plus 7 molecule of CoA. Omega alicyclic fatty acids. In numerous species of bacteria, omega alicyclic fatty acid are the foremost membrane fatty acids, classically enclosing an omega terminal propyl or butyryl cyclic group. The fatty acid synthase employed to build omega alicyclic fatty acid is also used to make membrane branch chain fatty acid. The regale of the cyclic carboxylic acid CoA esters is a lot superior to that of branch chain primers in bacteria with omega alicyclic fatty acids. The cyclic primer synthesis is not well known, but It has been recommended that procedure engrosses the conversion of sugars to succinic acid, followed by cyclohexyl carboxylic acid coesters that dole out as primers for omega alicyclic fatty acid synthesis. Synthesis of tuberculostearic acid. Tuberculostearic acid, chemically known as D10-methylstearic acid, is a saturated fatty acid well known to be produced by Mycobacterium species and two species of Streptomyces. Oleic acid, which is a monosaturated fatty acid, is the precursor that forms the tuberculostearic acid by the esterification to a phospholipid S-adenosylmethionine that donates a methyl group to the double bond of oleic acid. The methylation results in the formation of intermediate 10-methylene octadecanoyl, which on successive reduction of the residue with NADPH as a cofactor results in the formation of 10-methyl stearic acid. The fatty acid synthase, the charismatic enzyme complex. In humans, fatty acid synthase is encoded by the FAS gene. It is a multi-enzyme protein that catalyzes fatty acid synthesis. Fatty acid synthase is not a solitary enzyme, though a whole enzymatic system composed of two identical 272 kD multifunctional polypeptide where substrates are handed from one functional domain to the subsequent domain, catalyzing the synthesis of palmitate from acetyl-CoA and malonyl-CoA in the charisma of cofactor NADPH. The metabolic function and classes include that fatty acid or aliphatic acid essential to energy formation and storage, cellular structure and as an intermediate in the hormone biosynthesis, and other biologically significant molecules. They are synthesized from acetyl-CoA and malonyl-CoA by series of decarboxylative glycine condensation reactions. Subsequent to every round of elongation, the beta-keto group is abridged to the saturated carbon chain by the chronological action of a keto reductase, dehydratase and enol reductase. The budding fatty acid chain is carried among these active sites while fastened covalently to the phosphopantethylene prosthetic group of an acyl carrier protein, followed by its release through the accomplishment of a thioesterase abreast of reaching 16 carbon chain length, that is of palmitic acid. There are two major classes of fatty acid synthase, the type 1 and the type 2. The type 1 system make the most of a single large multifunctional polypeptide and are widespread to the both mammals and fungi. This system is also found in CMN group of bacteria, where it produces palmitic acid and collaborates with FAS2 system to produce a superior range of lipid products. On the other hand, type 2 system is present in archaea and bacteria and is exemplified by the use of distinct monofunctional enzymes for fatty acid synthase. Investigators are searching for the inhibitors of this pathway FAS2 as probable antibiotics. The mechanism of elongation and reduction in both FAS1 and FAS2 systems is similar because the domains of these two enzymes are mostly homologous to their domain counterparts in FAS1 multi-enzyme. The mechanism of elongation and reduction in both FAS1 and FAS2 is similar because the domains of the FAS2 enzymes are mostly homologous to their domain counterparts in FAS1 multi-enzyme polypeptide. Conversely, the disparity in the organization of the enzyme that is coordinated in FAS1 and distinct in FAS2 gives rise to a lot of essential biochemical differences. The evolutionary history of FAS is very much disheveled. The regulation and clinical significance of fatty acid synthase system is also important. The fatty acid synthase system is transcriptionally regulated and it is achieved through upstream stimulatory factors, also called as USF, which exists in two isoforms, USF1 and USF2, as well as the sterol regulatory element binding protein 1C, which is also called as SREP1C. Liver X receptor LXRs alters the expression of SREP1C, and acyl fluoroglucinols isolated from the fun's Dryopteris crassirhizoma demonstrated a fatty acid synthase inhibitory activity, which itself explains the regulatory part of the fatty acid synthase system. Genes that codes for fatty acid synthase have been examined as a possible oncogene, which has been revealed to be upregulated in breast cancers. Now, endogenous ligands for the nuclear receptor PPAR alpha is another important regulatory factor which has a role in fatty acid synthesis. These are the possible drug target for treating the metabolic syndrome. So students, today we have learned about the synthesis of the branch chain fatty acids and how this BCKA decarboxylase activity affects the synthesis of the fatty acids, as well as the regulatory and the clinical significance part of the fatty acid synthase system. Thank you.